Our next speaker is Dr. Billy Joel Almarinez. He is a full-time associate professor at the biology department de La Salle University. His PhD was obtained from the same institution under the Biological Control Research Unit of the Center for Natural Science and Environment Research. He has been working for several years now on the auspices of people like Dr. Barion, Dr. Amalin, on the biology and ecology of an endoparasitoid Compariella caulanica of the coconut scale insect Aspidiotus rigidus. We're back to coconuts and its role as a biological control agent for this species. Billy is a proud graduate of the Philippine Normal University and an example of how trained teachers who are encouraged to become trained researchers are the best in inspiring students to pursue science and engineering careers. So I'm moving it over to you, Sir Billy.
Thank you very much for that kind introduction, Ma'am Chi. A very pleasant day to everyone. I am Billy Almarinez from the Biology Department and the Biological Control Research Unit of De La Salle University. I'm here to talk about biological control, especially in this state of the new normal. Okay, we all know that the COVID-19 pandemic has brought about this new normal where we are discouraged from unnecessarily going out of our homes or our facilities and encouraging us to do more of our work indoors. And because of these quarantine measures that have limited the movement of people outdoors and in the field, there could be certain issues regarding agriculture and agricultural workers. So I believe that agricultural workers are not exempted from this new normal as well as from quarantine measures. So we have to be able to come up with ways to help them minimize the need to go out unnecessarily, okay, in their fields. Hence, I am here to introduce and propose biological control as a strategy for pest management that I think would require less human intervention, especially when it is compared to the more traditional chemical control. Biological control employs natural enemies of a pest, and these natural enemies could be predators or parasites or parasitoids or even disease-causing organisms found in nature. And these natural enemies may be able to control the population of the pest down to levels wherein the pest can no longer cause any damage to the productivity of our crops. Okay. Now, a noteworthy and I should say successful case of biological control in the Philippines would be that of Compariella calawanica against the cocolizap. The cocolizap or Aspidiotus rigidus caused an outbreak in southern Tagalog in the island of Luzon, and this outbreak caused or resulted in massive devastation of coconut plantations in the provinces of Cavite, Batangas, Laguna, some parts of Quezon, and a few parts of Rizal. And the cocolizap or Aspidiotus rigidus can impair the photosynthetic capability of the host coconut palm because when these insects feed on the foliage of the host plant, okay, the saliva of these insects destroys the chlorophyll and the chloroplasts of the tissues of the foliage. Additionally, transpiration and water transport in the affected host plant can be impaired because in cases of very heavy infestation, severe infestation, and very dense colonies are encrusting on the underside of the coconut leaflets, the insects tend to block the stomata through which transpiration occurs. So if unabated, severe infestation by the cocolisa can lead to the death of the host coconut palm. The government, through the Philippine Coconut Authority, implemented control measures, including frank injection of the systemic insecticide dinotefuran. They also attempted biological control. Unfortunately, it did not work because what they mass reared and released were natural enemies, including predators of another species of cocolisa, not Aspidiotus rigidus, but Aspidiotus destructor. And these predators, like Chilocorus nigritus, while they feed on Aspidiotus destructor and are known to be effective biological control agents of Aspidiotus destructor, they do not feed on Aspidiotus rigidus. There are also claims that the cocolisa outbreak in southern Tagalog went down as a result of the Typhoon Ramasun or Typhoon Glenda that occurred sometime in July 2014. In our study site in Calauan, Laguna, in Hidden Valley Springs Resort, we observed that the coconut palms there were able to completely recover even without any intervention by the Philippine Coconut Authority, meaning there was no trunk injection of dinotefuran, nor release of natural enemies of Aspidiotus destructor in Hidden Valley Springs Resort. Nevertheless, the coconut palms were able to recover, and we believe that such recovery, not just in Calauan, Laguna, but in the other parts of southern Tagalog where the outbreak occurred, okay, we believe that the insect that we discovered in Calauan, Laguna, Comperiella calawanica, contributed greatly to that recovery. Comperiella calawanica was confirmed through our laboratory and field studies to be a very effective natural enemy of cocolisa or Aspidiotus rigidus. And during the time of our discovery, it was a new species and we named it after the type locality or Calauan, Laguna, where we found the first specimens. And we also found that Comperiella calawanica exhibits characteristics that would suggest that it could be an effective biological control agent against its host, Aspidiotus rigidus, which is now considered a major pest of coconut here in the Philippines. In the field, we were able to record, parallel to the decline in the abundance of Aspidiotus rigidus in southern Tagalog, the remarkable recovery of what used to be heavily infested coconut palms in our study sites in southern Tagalog between 2014 and 2016. And we also were able to record very high parasitization levels by Comperiella calawanica, as indicated by those percentages. Okay, very high parasitization on Aspidiotus rigidus by Comperiella calawanica, close to 100% and more than 50%. That's why we believe that parasitism by Comperiella calawanica was a major contributor to the recovery of the coconut palms in southern Tagalog. And from our researches in the field and in the laboratory, and through the help of funding coming from DOST Picard and De La Salle University, we were able to develop technologies for mass rearing and strategic release of Comperiella calawanica as biological control agent against Aspidiotus rigidus or cocolisa. So Through our government-funded project, we were able to develop protocols for laboratory-based as well as outdoor-based rearing of Comperiella calawanica for biological control of Aspidiotus rigidus in areas of new invasion. Okay, so if we confirm invasion or infestation by Aspidiotus rigidus in areas where Comperiella calawanica is not yet present, we intended to release the mass-reared Comperiella calawanica from our facilities in Laguna to those areas. And in late 2016, we were able to identify and confirm such an area of new invasion by Aspidiotus rigidus in Zamboanga City. And upon our confirmation of Aspidiotus rigidus infestation in Zamboanga City, we brought to and released mass-reared Comperiella calawanica from our facilities in Laguna to Zamboanga City. And we observed that the Comperiella calawanica we mass-reared in Laguna readily attacked Aspidiotus rigidus in Zamboanga City. And in a couple of months, the Comperiella calawanica were able to establish populations and spread in affected areas of Zamboanga Peninsula, not just Zamboanga City. Okay? And the Philippine Coconut Authority has now adopted Comperiella calawanica as biological control agent against Cocolizap or Aspidiotus rigidus. They have adopted the technologies that we were able to develop, including strategic release methods for Comperiella calawanica. And in less than a year, recovery of what used to be heavily infested coconut palms was recorded in Zamboanga City as well as in other areas in Zamboanga Peninsula where we released Comperiella calawanica. Okay? So there was no major climatic disturbance in Zamboanga Peninsula from 2017 to 2020, so we cannot say that the typhoon aided in the recovery of the coconut palms. So we see that there really is promise in biological control as a pest management strategy. Now, we have been developing biological control technologies and strategies, not just for coconut, but also for cacao. Okay? For major pests, insect pests of cacao like the cacao mirid bug, we have already identified candidate biological control agents like some species of spiders as well as some species of ants that readily attack and prey on the cacao mirid bug. So there is promise in biological control, especially in this time of the new normal. But in order for us to be able to develop biological control technologies, we need to do basic and applied researches that need support from government agencies, from funding agencies, even from industry, and from the general public. That we need to convince that what we have been doing in our laboratories and in the field are worthwhile and are bearing fruits that are beneficial to our society. But in order for us to convince the general public, we have to educate them. We have to communicate effectively to them the results of our researches, the technologies that we have developed from them, as well as the implications to our society. Okay? So with that, I would like to thank our funding agencies, the Department of Science and Technology, PICARD, De La Salle University, and also the Philippine Coconut Authority for their invaluable assistance in our fieldwork in Zamboanga City, most notably Sir Ramon Rivera. Okay, thank you very much for your kind attention. Animo Lasal, all glory belongs to our Lord God.
Thank you, Sir Billy. That was a very, very interesting discussion. Actually, we're getting questions from previous talks and not the current talk, so we're just going to move them to the end. Sir Billy, just quick question. If you look at this technology of finding biological control agents, it seems very amazing. Do you know, you said you've worked with coconut and cacao. What do you think would be a good strategy in terms of developing work in the lab that can be used for this, when things like this happen? And what are the organisms that we should be focusing on in terms of the developing of biological control agents?
Thank you for that, for those questions, Ma'am Menchi. And by the way, before I answer, I would like to acknowledge Dr. Divina Amalin, who has been very instrumental as the director of TLSU Sensor, as well as the former lead scientist of Biological Control Research Unit. Uh, when it comes to discovering new new candidate biological control agents in the field, I guess nowadays that would be the challenge because of the new normal, because of the quarantine measures. But uh, for those uh, for those pests with documented natural enemy complexes already, I think the first step that we can do is to know if these are present in the Philippines, even if we're not going out in the field. Perhaps we can derive from reports, from published reports, if these natural enemies occur. And by natural enemies, I'm not just referring uh, to predators or parasitoids, but also pathogens, okay, that can cause fatality to the pests, but not any harm on human beings or other animals. So when it comes to laboratory work, when it comes to to discovery of new candidate biological control agents, unfortunately nowadays, and I think that's one sad thing that I can relate to right now, because personally I cannot go to our laboratory. Okay, uh, but when it comes to biological control agents that we may already have in our laboratories, then fortunately we have people in our laboratory that continue our work. Okay. Actually, there's also an avenue for discovery of biological control agents in uh, in microbiology, because pathogens, entomopathogens, could be fungi, could be bacteria, uh, and I think they're easier to handle in the laboratory compared to
Live.
uh, live insects or spiders. And uh, so unfortunately, that's how the new
Yeah.
normal limits us. But when it comes to opportunities for what we already have right now, the biological control technologies and biologically based pest management strategies that we have already developed, there is promise not just in directly applying these technologies to agriculture, but also in terms of partnering with industries. And I think Sir Rene, I think I saw Sir Rene type a comment about the possibility of partnering with industry. Actually, it is a promising endeavor, Mamenchi, because actually in the US, uh, biological control agents are those that are already being used widely in
Mm.
agricultural areas. They're actually no longer produced by the scientific facilities, but they're already produced by industries.
Wow. All right.
So there's also economic promise in biological control.
Yeah.
Uh, as long as we are able to partner with industry, and we have to communicate, really communicate this
Yeah.
uh, to the industry, because I'm sure not many people know about biological
Yes.
control.
Yes. Definitely. Thank you for that, Sir Billy. That was an amazing discussion. And for students, now systematics Ecology, these are not foregone topics, and they can actually be melded very easily with the kind of things that industry will need.